Welcome back to Virology. This is a special Sunday evening episode. And today we are going to talk about therapeutic viruses. This is our last lecture for this 2024 virology course. And this is a fitting way to end this lecture because we're going to take everything we learned and use it to treat diseases. We're going to make therapeutic viruses. This is not a lecture we had in the very beginnings of this course. This is pretty new. It happened because the technology developed in order to do what we're going to talk about today. So what are therapeutic viruses? We're going to talk about 4 different uses of viruses for therapeutic uses. First, phage therapy for bacterial infections. Second, gene therapy, that is delivering a gene to patients who lack the gene or have defective versions of the gene. We will talk about using vectors to deliver antigens, viral vaccines, and finally, viral oncotherapy, treating cancers with viruses. A key for vector development. So what we're going to do in this lecture is we are going to take the virus genome and modify it to suit our purposes. And to do that, you need an infectious DNA copy of the viral DNA. And the very first infectious DNA of an animal virus was that of poliovirus, shown on this slide. We've seen this before. I'm very proud that this is the experiment I did as a postdoc way back in 1980. And this is essential for every virus that we have infectious DNA technology where you can take the genome and make a DNA copy in a plasmid, say, and then either introduce that plasmid into cells or make RNA in vitro, and then the RNA is put in cells, and in each case you get infectious virus. This is completely essential for all the things we're going to talk about today. So phage therapy first. After discovering phages in 2015, Darrell pursued their use to treat bacterial infections. He co-founded the Eliava Institute in 1923. That's in Tbilisi, Georgia, and it's active to this day. It produced phages for antibacterial therapy during World War II. You can still find these kits that the soldiers got, which have vials of phages. So if they had a gastrointestinal bacterial infection, they could drink it. And I don't know how effective they were, but they were given them. Unfortunately, in the 1930s, the introduction of antimicrobial drugs dampened enthusiasm for phage therapy because they were much easier to make. And so for many years, not many people worked on phage therapy. The Eliava Institute continued to do so. But now, as you know, we have widespread antimicrobial resistance, antibiotic resistance, and this has revitalized interest in phages to treat bacterial infections. The principles of phage therapy is that you use the phage to kill specific bacteria that are, say, causing your disease. You don't want to destroy our normal flora. Fortunately, phages can be very specific to the bacterium, and that's not a problem. And so let's say if someone has an infection, you have to identify, of course, what the bacterium is that's causing the infection. You have to make sure you have a phage that can kill that bacterium before the treatment. So this is not easy. It's not straightforward. You have to almost design the phage for every patient, although this is starting to be done, and it looks like it might be possible. We have already licensed phage lysins. So lysins are the proteins made in phage-infected bacteria that are responsible for lysing the cell. And these have been licensed for surface decontamination, say in food preparation areas, you can spray them with the lysins. And what they do is they rupture the peptidoglycan in the membrane so bacteria can have besides a membrane, they have a peptidoglycan, and the lysins can break both, and that causes the bacteria to rupture. And there's another one called, there is a phage actually called agriphage, which was approved by the EPA in 2005 to treat a plant disease called bacterial tomato canker. So that's an improved phage treatment for a plant disease. And then List Shield is a listeria phage approved by the FDA for contamination of meat and poultry. And this was the first to get this designation, generally regarded as safe, which is really important to get anything approved for human therapy. So listeria is a bacteria that causes foodborne illness. And so this is used to decrease the bacterial load on the surface of meats. So that's been approved also. Here's some successes in phage therapy that have been published recently. This one here on the very top left, phage treatment of an aortic graft infected with Pseudomonas. That's quite interesting. This gentleman had an aortic graft installed and it got infected. So they decided to treat it with phage. It was resistant to antimicrobials. It was infected with Pseudomonas aeruginosa. And so they did treat they opened the patient up and they poured phage on the graft. Apparently it was so fragile that they didn't risk injecting it. So they just poured it on the graft and sewed him back up. And he actually was cured of his infected graft. And about a year later, the patient died of other causes, actually on the day that this paper was published here. And we talked about this paper on Twivo 44, below this Nature Medicine, engineered phages for treatment of a patient with disseminated drug-resistant Mycobacterium abscessus. So this was an immunosuppressed patient that was infected with this mycobacterium. They treated him with phages that were selected to be able to infect that mycobacterium. And during the course of the treatment, he got better. The bacteria became resistant to the phage, so they re-engineered new phage to kill those bacteria. And so a nice example of how you can change the therapy during treatment. And then finally, antimicrobial agents and chemotherapy here on the right. Development and use of personalized phage-based therapeutic cocktails to treat a patient with disseminated resistant Acinetobacter baumannii infection. There's a very famous case of a man. Let's see if he's an author on this paper, actually. No, he's not. But his wife here, Stephanie Strathdy. So he got a bad infection when he was visiting, I think, Egypt. And it became multi-drug resistant. And so she Googled phage therapy and found some people in Texas and the U.S. Navy who had phages to match his infection. They treated him and he's recovered. He's fine today. He was about to die. And she has written a book about that. I interviewed her actually at South by Southwest. I think I put the link to that in a previous lecture. And we talked with the group at Texas A&M who engineered the phage on TWIV 502. So we've been fortunate to be able to talk to a lot of people. So Strathdy is very bullish on this technology, as you can imagine, but it doesn't work for everyone. There's still things to overcome. For example, making antibodies. So in this paper, you see potent antibody-mediated neutralization limits bacteriophage treatment of a pulmonary Mycobacterium abscessus infection. So this is a case where an individual was treated with a bacteriophage for a systemic infection, again, Mycobacterium abscessus. And the patient made antibodies to the phage, and the phage no longer could work. So this has happened again subsequently to this, a woman who had an intestinal, actually a systemic infection that originated in her intestine. She made antibodies that made the phage not useful. So the key probably is to make new phage preparations or have them ready when you have ones that are resistant. But as you can see, this is an evolving field, and there's a lot more work to do, but very exciting, I think. Let's talk now about using animal virus vectors to treat diseases. First we're going to talk about adenoviruses. Remember, these are large DNA-containing viruses, icosahedral capsids, 36,000 base pairs of DNA. And this has been used for various vector usage. Some of the advantages, these viruses efficiently infect postmitotic cells. That means cells that are not dividing. And you know why? Because when the virus infects non-dividing cells, it makes them divide, right? By making T antigen. Fast onset of gene expression after infection. The virus remains episomal, so there's very minimal risk of insertion mutagenesis. Big capacity, easy to make pure concentrated preps, lots of human and animal serotypes, but the drawbacks are you make antibodies to it, and this is an acute infection. So if you need a protein made for a short period of time, say in a vaccine, this is great, but not if you need persistent expression. The very first generation adenovirus vectors were E1 and E3 deleted. So the first line here in blue is the wild-type adenovirus genome, DNA represented as a bar with the inverted terminal repeats, the packaging sequence on the left. This is why I say this lecture taps into everything you learned in this course. If you remember, Psi, packaging sequences, inverted terminal repeats. Okay, so this vector is deleted for E1. E1 encodes T antigens, right? Which are needed to kick the cell into division, but this vector is not going to replicate, so it doesn't matter. And then E3 is got immunomodulatory proteins in it. Actually, we will grow this vector in the presence of cells that produce the E1 protein, so it will reproduce to high titers, but when it infects the cell, it's not going to replicate. It's just going to do gene expression. Second generation, E1 and E3 deleted, as we just saw in first, but then E2 and E4 were deleted because those were found not to be essential. And then the third generation vectors are called gutless vectors. All the genes are deleted, and all you have are the two inverted terminal repeats and the packaging sequence. So here on the top is a diagram of the vector. You have the left inverted terminal repeat, the packaging sequences, which is about 400 or 500 base pairs, and then the right inverted terminal repeat. And you stick your transgene in here, and there's a promoter shown with the red arrow here that drives the gene expression. This needs a helper virus to package it, and the helper virus itself is E1 deleted. And here is a very clever scheme for packaging this vector. So here again at the top is the vector derived from wild-type adenovirus. The helper virus has to provide capsid proteins, right? And so this is the helper genome here at the bottom, and it has 2 LOXP sites in the packaging region at the left hand of the genome. And it's also deleted for the E1 region, so this vector is not going to replicate. So the LOXP sites are targets for the Cre recombinase. So when this DNA is put into cells, these are producing E1, these cells, so that this DNA can replicate. The recombinase, which is also produced in the cells, will cut out the packaging sequences from this vector, so the vector will not be packaged, but it will make proteins that will the helper will not be packaged. But the helper will make capsid proteins that will package the vector shown at the top here. So that's done because you don't want the helper DNA to be packaged. Adeno-associated virus vectors are very popular. Remember, these are single-stranded DNA viruses with the funny terminal repeats at either end. They encode very few proteins. The rep protein, remember, which is basically an origin binding protein and endonuclease, and then the capsid proteins down here. These have been modified to make vectors, and the advantage is you get long-term expression of these viruses, and there are many serotypes. And so in practice, what you do is you take the genome here at the top, which has inverted terminal repeats, the rep gene, and the cap gene. You delete rep and cap, and you put a transgene in there. You make sure there's a promoter to drive transgene transcription. You then introduce that construct into cells along with a rep cap plasmid so that the DNA will replicate and it will get packaged, and adenovirus helper functions. Remember, this is a helper-dependent virus, adenovirus-associated virus. And those are E1A, E1B, E2, E4, and VA RNA. And out comes recombinant adeno-associated virus. It's so cool. Now, these genomes persist in cells as episomes. Now, if you infect cells with AAV without a helper virus, the DNA will integrate. But that requires the rep proteins, and we don't make those in vector-infected cells. We have deleted the rep genes, which was on the previous slide. So this vector comes into the cell. It's got the ITRs and the transgene. What it does, and there are multiple copies of that, they recombine to form circles. And those circles persist for many, many years in cells. As you'll see, we have some amazing stories of successful treatment of certain diseases with these vectors. They persist. This was totally unexpected. We didn't know they would persist. We knew they integrated, but we didn't want that to happen because, when DNA integrates randomly into cells, it can cause problems. And so this just shows that you have to be curious. You have to just pursue things and see where they lead. Which technology is indispensable for the production of therapeutic viruses? A, X-ray crystallography. B, high throughput genome sequencing. C, synthesis of infectious DNA copies of viral genomes, plaque assay, or immunofluorescence. You can't see the results, but I'll tell you. It's 100%. Synthesis of infectious DNA copies of viral genomes. So all of you got synthesis of infectious DNA copies. That's essential for producing therapeutic viruses. All the other things are not essential for that. So that's like number 9, right? You guys have a record in the most 100% answers. Retrovirus vectors are very popular as well. These are based on the fact that if you just produce the viral GAG-POL protein, and on a separate plasmid, the envelope protein, you put those two plasmids into cells, you will get particles made without the viral genome. So the GAG-POL makes the capsid and the membrane protein and drives the assembly of the envelope with the spikes. And it also will have reverse transcriptase and integrase in the capsid. And then the envelope, of course, drives the spike protein in the membrane. And so if you put a third plasmid in with a transgene, you can get that incorporated into the retrovirus particle. The key is, of course, to have this green sequence here, which is the packaging sequence. So you put the packaging sequence in the transgene plasmid, it'll make an RNA that gets packaged into the particle. So you see how our understanding of the molecular biology of viruses plays into these therapeutic applications. This is a theme we're going to see throughout. That's why I say this lecture builds on everything that you learned in this course. So these vectors are typically based on lentiviruses, HIV-1 in particular, or other retroviruses. HIV is cool because it can infect non-dividing cells. The DNA will integrate right into the cell genome. So you get long-term expression from the provirus. You can put up to 8 kb transgene inserts. However, because the provirus is in the DNA, there's a possibility for insertional immunogenesis, right? So the provirus could go next to an oncogene and activate it, and that would cause cancer. So what we did, we actually found that, unfortunately, in a clinical trial. So now we inactivate the 3' LTR promoter so that doesn't happen. And we also pseudotype these viruses with the glycoprotein of VSVG because it infects many different cells. So we don't actually use HIV or other retrovirus glycoproteins. We use the glycoprotein of VSV. We use poxvirus vectors. You may know that some of the COVID vaccines used poxvirus vector. The backbone is a virus called modified vaccinia virus Ankara. This was originally produced as an alternative smallpox vaccine and, in fact, is part of the U.S. strategic national stockpile. Did you know we have a stockpile of various vaccines and antivirals to be used in case of an outbreak? An Mpox outbreak was treated with this vaccine and also with antivirals that were produced. There's also a vaccine against H5N1 influenza virus in the strategic national stockpile. This is a replication deficient vector. This virus is infectious in avian cells, but not in mammalian cells. And that's because the virus was passaged extensively in chicken cells, and there's an assembly block latent infection. So it will get into mammalian cells and replicate, do gene expression, but it won't make infectious virus. So you can work with it under BSL-1, very easy lab conditions, it has a big capacity. And we've also used another pox virus as vectors, canary pox virus, which you'll hear be mentioned today. So here's how you make recombinants with MVA. So you're going to take your DNA that you want to incorporate into the virus. Those sequences are shown in red here. And you put it in a plasmid, a bacterial plasmid. And then on either side of the vector DNA, the insert DNA, you put sequences from MVA, and that's going to stimulate recombination. So you introduce the plasmid into cells by transfection. You then infect with MVA. The DNA will recombine into the viral DNA, and out will come recombinants. You can see on this picture here, this viral DNA, which is mostly red, has the insert there. Sorry, it's mostly blue, has the red insert there. So these are relatively easy to make. And we use VSV as a vector. Remember, this is a negative strand RNA virus with an envelope. The negative strand genome is shown at the top. It encodes 5 mRNAs encoding the various proteins. And the strategy that's used is to replace the spike gene, the G protein gene, with that of your transgene. And of course, in this case, you have to replace it with another protein that's going to be a spike, right? Because you need a spike to infect cells. And so this was attempted for COVID vaccines. It's been done for Ebola virus vaccines and others. And what you do is you have 4 plasmids encoding the viral proteins NPL, and then your other plasmid has the viral RNA genome with the transgene inserted. And you put 4 plasmids into cells. These plasmids have T7 promoters to make mRNA. So you also infect with vaccinia virus encoding T7 RNA polymerase, and out comes infectious VSV. Flaviviruses can be used as vectors. These are plus-stranded RNA viruses like dengue and West Nile. And so this is actually the genome of yellow fever vaccine virus. This is a vaccine virus that's been put in over 500 million people. It's safe. And so we modify it to carry other flaviviral antigens. We take the structural proteins here on the left, PRM and E, and we replace them. In the one example I'll tell you, we replace them with the structural proteins of dengue virus. And you take a DNA encoding this construct, basically yellow fever genome with dengue virus proteins. You make RNA transcripts in vitro, transfect them into cells, and out come your recombinant virus that can be used for immunization. And this vaccine is called Dengvaxia, has been licensed in many countries. We use alpha viruses, Semliki forest virus. These are plus-stranded RNA viruses, which have a subgenomic mRNA. We talked about these because that's what's unusual about those. And so what you can do is replace the structural proteins with those of another alpha virus. So again, it has to be an alpha virus because you need to make a capsid, but you can take advantage of a good replicating alpha virus to put other alpha virus proteins in it. And again, you replace the structural genes in a DNA copy of the genome, you make RNA in vitro, transfect it into cells, and out comes your recombinant virus. And finally, Newcastle disease virus. This is a paramyxovirus with a negative strand genome. It's an envelope virus, which is a pathogen of chickens. And so farmers have been using this to immunize chickens for many years. But now it's been modified to carry a transgene. And typically what you do is you insert the transgene between the P and the M proteins here. So you make a plasmid with the transgene inserted into the viral RNA. You have 3 other plasmids, very much like VSV, and you transfect all 4 in with T7 produced by vaccinia virus, and out comes Newcastle disease virus with carrying a transgene. And typically, it's another spike protein from some other virus. So here are some licensed vaccines that use viral vectors. Ervebo is a VSV carrying the glycoprotein of Zaire Ebola virus. This has been licensed and it's used extensively in Africa. Dengvaxia, the structural region of 4 dengue serotypes, was put into the yellow fever vaccine vector. We have a yellow fever vaccine vector Japanese encephalitis virus for humans, and also a West Nile virus one for horses. As I mentioned, we have an H5, or maybe I didn't mention it. With Newcastle disease virus, we have a vaccine carrying the hemagglutinin of avian influenza virus. So you can immunize chickens with this, and they'll get both immunization against Newcastle disease virus and H5 avian influenza virus. You may remember during COVID, an American company, Janssen, made an adeno-26 vector carrying the spike gene of SARS-CoV-2. This turned out to cause severe clotting disorders and rarely, but severe. And so that's no longer used, was withdrawn. ChAdOx vector is a adenovirus serotype 26 chimpanzee adenovirus vector carrying the spike protein of SARS-CoV-2. It's called Vaxzevria, and it was used extensively in Europe and many other countries, still is. And then in Russia, they made a vaccine called Sputnik, which comprises a 2-dose vaccine, one with adenovirus 5 and one with adeno-26, both carrying the spike proteins of SARS-CoV-2. Now, a new entry into the vaccine field is called self-amplifying mRNA vaccines. Now, you may know, and I think we talked about this, the SARS-CoV-2 mRNA vaccines were produced by taking an mRNA, encoding the spike protein, and putting it in a lipid nanoparticle. This gets taken up into cells, and the mRNA is translated into protein, which is then taken and given to lymph nodes for antigen presentation. The self-amplifying mRNA vaccines are similar. They're an mRNA delivered in a lipid nanoparticle, but the RNA replicates. So you get much more protein with a lower dose of vaccine. So how does this work? So we talked just briefly about recombinant RNA viruses like flaviviruses, where you put a gene of interest in the genome. And these recombinant viruses can replicate, and of course their RNA is amplified. But replicon RNA and mRNA vaccines are shown at the top. It's just an mRNA, doesn't get amplified, it doesn't propagate. But you can make self-amplifying mRNAs. Here's one based on alpha viruses. Here's the gene of interest, and it's like a spike protein, for example. And up here are the RNA polymerase genes of the virus. So when you infect cells, this RNA will replicate. You don't get virus particles made, right? It doesn't propagate like the recombinant virus does, but you get self-amplification. And we also have flavivirus-based replicon RNAs, where again, you have your gene of interest, and then the blue are the self-amplifying genes, which means the RNA polymerase and accessory proteins. So I think we should keep an eye on these, because you can give lower doses of these self-amplifying mRNA vaccines compared to the traditional mRNA vaccines, and no infectious viruses are produced. All right, next we're going to talk about gene therapy for monogenic diseases. What is a monogenic disease? It's caused by a mutation in one gene. All right, so you have a gene, a single mutation, and you can look at the inheritance pattern. It could be dominant or recessive. These occur, there are over 6,000 different monogenic diseases, and they occur in 1 in 200 live births, really a lot. So these are amenable to viral gene therapy because we can give patients a correct form of the gene. And currently there are over 1,800 clinical trials in process for different vectors for monogenic diseases. There are a lot of these, and I just want to give you a sense for a few of them here. Here's the disease, the defect, the incidence, and vectors that are being explored to treat them. So, for example, SCID, severe combined immunodeficiency, where people don't have T or B or NK cells. And there are two different kinds of defects, either an ADA gene or a common cytokine receptor gamma chain. And those can be corrected by putting the gene in a retrovirus vector. And those are actually licensed, as I'll tell you in a moment. Hemophilias, hemoglobinopathies, retinal diseases, etc. All of these are being tested with different vectors to try and correct the deficiency. This is a graph of the number of gene therapy trials from 1990 to 2022. You can see it increases every year, currently 220 different ones in trial. And this is a very, very exciting field. There are two general ways that this works. First, you could put your gene into a virus, the correct gene, and simply infect the patient with it. Now that's not great, because when you inject, say, virus into the bloodstream, it goes immediately to the liver and it's trapped there. So if you want to put a gene in the liver, that's the place to go. The better way is to do cell-based delivery. Say we take T cells from a patient. We're going to talk about this later. And we infect them with a viral vector. In this case, it's making a new antigen called a CAR antigen. And then we put the T cells back in the patient. Or we can take stem cells. We can take stem cells from the patient, or we can use embryonic stem cells or get a stem cell bank sample. We can introduce the virus into those and introduce those back in the patient. The stem cells, of course, can become many other cell types producing the therapeutic protein. So let's look now at some examples of gene therapy over the years. This is one of the first human viral gene therapy trials, 1993. A male with cystic fibrosis. This is a disease where you have mutations in the cystic fibrosis transporter gene, which predisposes you to severe lung infections. And so you have a defect in the gene. So what we want to do is but give the wild type copy of the gene. And so this was a first generation adenovirus vector, E1, E3 deleted, with the DNA encoding wild type CFTR. And it was a lot of virus, 2 times 10 to the 8th PFU administered to the airway epithelium. So you can see the patient here, he's undergoing endoscopy, bronchoscopy, and they're spraying the adenovirus into the airway. And here's a sample of his lung cells, pre-therapy, stained with an antibody to the protein, to the CFTR protein. And you can see 4 days post-therapy, his cells are making the CFTR. And this is a graph showing the ratio of exogenous to endogenous CFTR. You can see in the first administration of vector, he made a burst of vector-encoded protein, and that went down. And then he got a second one. And then on the third time, it didn't work anymore. And so this is because he's making antibodies to the vector. And so this taught us early on that we need to do something about that. The real issue with this trial is that it's an acute infection. It's over in 2 weeks. And that's not good enough to correct the defect. So you have to have more long-term approaches. Many clinical trials were done after that one, but in 1999, there was a serious setback. This is the first person to die in a gene therapy trial, very well-publicized event. His name was Jesse Gelsinger. He was an 18-year-old whose father volunteered him for this trial, and it was to correct deficiency in ornithine transcarbamylase. This is an X-linked disease that leads to the accumulation of ammonia and glutamate in the blood. And patients with the most severe deficiencies have declining cognitive ability and premature death. So the idea here was to give an adenovirus vector with the normal OTC gene. They started a trial at the University of Pennsylvania. They had 18 persons enrolled. Jesse was the 18th person to receive the therapy. 4 days after he got it, he died of a massive immunoinflammation, immune response, and multiple organ failure. And an examination of this revealed that multiple rules of conduct were broken at the University of Pennsylvania. They didn't do a lot of things that they should have. All of the trials were stopped until this could be taken care of. So this was an unfortunate event that didn't have to happen, but made the field realize you have to pay attention to the details. You can read a lot about this online. And in fact, his father has written extensively about this and how he feels about it. Another disease, X-linked SCID, severe combined immunodeficiency, where you have no TB or NK cells. These are present. These are mutations present in the IL-2 receptor gamma chain. And this is the gene shown here. And these are mutations in various patients that have been observed. And so a trial was designed, 2 trials actually, in London and Paris, where they gave infants a retrovirus containing the normal IL-2 receptor gamma chain. So what they did is they took bone marrow hematopoietic precursor cells from the babies. They infected them with the retrovirus in cells outside the patient in culture, and then they transplanted them back into the patients. These patients actually had good recovery of their immune function, but many years later, in fact, 4 out of 9 patients in Paris and 1 in London developed T-cell leukemia 3 to 6 years after treatment. And this was recognized to be a problem with the retrovirus vector, as I'll show you in a minute. And so at the time, there were 27 trials with retroviral vectors ongoing. They were all halted to fix what was going on. And what they used was a non-lentivirus vector, Maloney-Murine leukemia virus vector. And it turned out that this integrated into the patient's DNA and activated oncogenes. So here, for example, are 2 patient integrations, patient 4 and 5. And this is the retroviral, the provirus, right, with the 2 LTRs and the therapeutic gene in the middle. These integrated near this gene called LMO2, which is an oncogene basically, and is involved in the various leukemias that these children got. And so the promoter is or the enhancer within the promoter is activating this gene when it shouldn't be activated. And so the solution was to make retrovirus vectors where the LTR is inactivated. So this doesn't happen anymore. So this has been fixed. And now a new trial was done a number of years ago. 8 infants with X-linked SCID were given bone marrow transplants with a lentiviral IL-2 receptor gamma chain into their bone marrow stem cells. And at 18 months, all of these babies had functional B and T cells. So this is really a good success story. And now the vectors don't cause cancers. Sickle cell disease is another that's amenable to gene therapy. Here you have a mutated beta globin gene. The protein misfolds and forms fibrils, and that causes the sickle-shaped deformation in the red blood cells. And we have 2 approaches to gene therapy. In one, we make a lentivirus that contains the normal anti-sickling beta globin protein, and we give that to patients. And again, we do that by explanting stem cells ex vivo and putting them back in the patient. And then more recently, a new approach using CRISPR-Cas9. So we deliver CRISPR-Cas9 to the patient's cells with viral vectors, and these cut out the mutated gene and replace it with a wild type copy of the gene. There are also a number of blindness diseases that are amenable to gene therapy. These are called inherited retinopathies. There's the eye, of course, it consists of, in the back of the eye, the retina consists of the retinal photoreceptor epithelium, and these are connected to the nerves by cones and rods. And there are many defects in these cells. It can be in the retinal pigment. Sorry, this is retinal pigment epithelium, the cones and the rods. This particular cell here, the ganglia itself, and each of these defects are shown as numbers here, and there are trials to correct them using different vectors: AAV, lentiviral, and adenovirus vectors. These are monogenic mutations in these cell types. They're untreatable blinding conditions. AAV has turned out to be the most promising, and what you do is you put the gene that's defective in the AAV, and you inject it directly into the retinal pigment epithelium. And so one of these mutations in the RPE65 gene is needed for photoreceptor function. In dogs, a single subretinal injection of AAV vector with the canine gene restores visual function. And this was then tested in humans and approved in December 2017 by the FDA. It's a drug called Luxturna. It's an AAV carrying the RPE65 gene. It's $900,000 for both eyes, but these patients can see again. And I spoke with the person who did much of the development, Catherine High from the University of Pennsylvania, on TWIV 350. So that was the first gene therapy to target a gene caused by mutations in a disease caused by mutations in a specific gene. Some other gene therapy successes. So we talked about SCID, that's approved for treatment. Cerebral adrenoleukodystrophy is a severe brain disease caused by mutations in the ABCD1 gene. This is approved as a drug called Skysona for $3 million, one treatment. Sickle cell disease, as I mentioned, there are two approaches to introducing the gene or to do a CRISPR-Cas edit. That's been approved. Beta thalassemia correction using a lentivirus carrying beta globin. Hemophilia A and B, which have two different clotting factors defective, both approved in AAV vectors for $3 million. Lipoprotein lipase deficiency in a AAV vector for $1 million. Spinal muscular atrophy, the SMN1 gene is delivered in an AAV vector. That's $2.25 million. And then Duchenne muscular dystrophy, the wild type gene is delivered in an AAV vector. And I put some of the prices here because this is obviously an area of contention. It's a lot of money. And, you know, the part of the problem is that some of these diseases are very rare, so companies have to recover their development costs. But it's also an assumption of how much you would pay to live or to see, right? And so not everyone can afford this. If you don't have good insurance, then you can't see. And I don't think that's very fair. So this needs to be fixed. All right, next question. Which of these viral vectors are not likely to be compromised by immune memory in humans? Newcastle disease virus, vaccinia virus, herpes simplex virus, or adenovirus? Okay, which is not likely to be compromised by immune memory in humans? Let's see what we did of your four. There's no winner. Half of you picked Newcastle disease virus, and half of you picked adenovirus. So vaccinia is a human virus, right? Herpes simplex is a human virus. So is adenovirus. So the answer is Newcastle disease virus. That's the one where if you use a vector made of Newcastle disease virus, we're not going to have problems with human immunity because it's chicken virus, right? Finally, the last part of today is viral oncotherapy, destroying tumors with viruses. And there's a couple of general approaches here. Two. First, some non-human animal viruses can replicate in human tumors. They don't replicate in humans, but when humans have tumors, they can reproduce in them, like myxoma virus. That's the virus used in Australia to try and eliminate the rabbits, or Seneca Valley virus. Or we can take human viruses and modify them to target and kill tumors, usually with immune enhancement. A number of years ago, Linda Coughlin, who was a virologist at the University of Maryland did an episode of Virus Watch about cancer-killing viruses. This story begins many years ago in the 40s and 50s, as you can see here. People had observed that certain individuals with virus infections, their cancers seemed to be doing better. So they did 4 trials here where they took a cancer and they treated it with a different virus. Now, this is before we regulated clinical trials. These are trials that would never be permitted today because there are no controls. The source of the virus, the route of administration varied from patient to patient. We didn't know the concentration of the virus. That varied from patient to patient. None of these yielded any useful information, but you can see they infected people with Hodgkin's lymphoma with hepatitis virus, and many of them developed hepatitis, and a few had transient responses to the tumor, and one died. We had people infected with West Nile virus, and you see many people got encephalitis. As you know, that's a common outcome of West Nile virus infection. Again, transient responses, Epstein-Barr virus. You know, if you give people viruses, they're going to get virus infections. They got mono, human adenovirus, transient tumor necrosis. But anyway, this gave the idea that maybe viruses could be useful. And so let me go through a few of the things we do to make viruses that target cancers. First of all, interferon defects are common in cancer cells. And so what we do is we introduce into the genomes of these oncolytic viruses mutations. So these are into genes that counter antiviral interferon defenses. So these include like in adenoviruses, E1B, in herpes viruses, ICP35, vaccinia, VSV, that are known to have suppressive effects on interferon. So because the tumor doesn't have interferon responses, the virus will reproduce there, but it won't reproduce in normal cells that have normal interferon responses. So this is a very clever way to make sure that the virus is selectively replicating in the tumor. All right, so that's one approach. So other approaches to make what we call tumor cell selective reproduction. Well, this again is what I just told you. VSV is sensitive to type 1 interferon, so it doesn't reproduce in most tissues, but tumors have a defect in type 1 interferon, so VSV will selectively reproduce in that. Many tumor cells express on the surface the protein CD46, which is a receptor for the measles virus vaccine strain. So that can be used to target infections to tumors. The herpes simplex glycoprotein D gene can be engineered to contain either IL-13 or antibodies against human epithelial growth factor receptor 2. Both proteins are present on gliomas and breast tumors, so you can basically target the viral vector to infect those cells. For adenovirus, we've inserted domains that recognize tumor antigens into the fiber. You remember the fiber, those proteins sticking out from the 5-fold axis that bind to receptors. So we can basically retarget the adenovirus to tumor antigens. We also can add adapters to adenoviruses to the fiber that retarget them to cancer cells. And finally, we introduce mutations in the E1 region of adenovirus. These are proteins, remember, E1A and E1B counter RB and P53 respectively. Many tumors have defects in those proteins. And so these mutant proteins, these mutant adenoviruses, will not reproduce in most normal cells, but they will reproduce in tumors that are deficient in RB or P53. We can do all kinds of post-entry targeting to make it the vector tumor cell specific. Here's one we call positive targeting. We basically make a virus that has some therapeutic gene, and you'll see examples of those in a moment, under the control of a promoter that does not work in normal cells, but works only in tumor cells. So when the virus infects a healthy cell, the gene won't be transcribed, but in a tumor cell it will be, and that can contribute to cell killing. We can also do negative targeting, which means we put a microRNA into the vector that is a sequence in the vector that's the target of a microRNA found in normal cells. And the microRNA is not present in tumor cells. And we can identify such microRNAs by simply doing northerns, looking at RNAs from various cells. So here's a particular two different microRNAs here, LET7A and MIR124A. And those are RNAs from different tissues. And you can see they vary in different tissues and tumors as well. So we can put a target of a microRNA that is not expressed in tumors. And so that restricts the replication in a normal cell and allows virus replication in a tumor cell. Some other strategies we can target easily tumors to certain tumors. We can easily target oncolytic viruses to specific tumors, as I've just told you. But how do we increase lysis? There are a number of strategies. Well, one is to put the P53 into adenoviruses, and that's a proapoptotic protein, if you remember. When P53 detects unscheduled DNA synthesis, it induces apoptosis. And so this is a strategy for killing tumor cells. We can incorporate cell genes that are prodrug convertases. Do you remember what those are? So a prodrug would be like acyclovir, shown at the bottom, which has to be phosphorylated by the thymidine kinase gene of herpes simplex 1 before it can get triphosphorylated and act as a chain terminator. So that's a prodrug convertase. And so you put that in the oncolytic virus, and then you give the patient something akin to acyclovir, and it will be activated just in the tumor. We can also add genes encoding viral proteins that allow cell spread of the virus. And so, for example, a fusion protein would allow cell spread in the tumor, and that would shield the oncolytic virus from neutralizing antibodies. So it keeps the virus going from tumor cell to tumor cell. Here's an example of arming cell vectors, oncolytic virus vectors, with prodrug convertases. So we can use thymidine kinase. This converts ganciclovir into ganciclovir triphosphate, and that causes DNA synthesis inhibition in tumor cells. Again, this is delivered by the oncolytic virus. We can put in the gene encoding cytosine deaminase. It converts 5-fluorocytosine to 5-fluorouracil, which is a chain terminator. And these stop DNA replication of tumor cells. So here's your uninfected tumor cell. The prodrug gets into them, but if it gets into a virus-infected tumor cell, the virus will convert the drug into the toxic metabolite. It will kill the tumor cell, and it will also diffuse to neighboring cells and kill them as well. Here's an example of this, a therapy called TOCA 511, which was meant to treat gliomas, right? Severe brain tumors. It's a murine leukemia virus with cytosine deaminase. So you give the virus intratumorally, right? You inject it into the tumor, and then you do it with 5-fluorocytosine at the same time. And so the cytosine deaminase is going to convert the 5-fluorocytosine into 5-fluorouracil, which in theory will kill the tumor. And this went into phase 1, 2, and 3 trials for gliomas. Unfortunately, was not effective and is no longer being pursued, but it illustrates the principle. A big part of viral oncotherapy is promoting anti-tumor immune responses. For example, the tumor environment is immunosuppressive. I told you it has innate defects, innate immunity defects, but also adaptive responses are disrupted. And so we can take advantage of that. For example, we can design vectors to, so in the tumor, the cell surface expression of receptors for checkpoint inhibitor ligands, PD-L1 and CTLA4 occurs, and this blocks T cell responses. So a tumor typically has excessive PD-L1 and CTLA4, not always, it depends on the tumor. And then when a cytotoxic T cell tries to kill the tumor cell, these block it. And that's the basis for monoclonal therapy against these receptors or their ligands. That helps get more effective tumor cell killing by the CTLs. But you can also incorporate the genes encoding these monoclonal antibodies. Again, the monoclonals against the ligands or their receptors. It's called checkpoint immunotherapy. And as I talked about earlier, the genomes of many viruses are modified to remove genes that block the type 1 interferon response. This will make them specific for the tumor, which is defective in the interferon response, but normal cells are not. And then finally, we include genes for immunostimulatory proteins like GM-CSF and chemokines that stimulate an immune response. So here's an example of incorporation of several of these features into a treatment, a herpes virus vector to treat melanoma. So this is a herpes virus called, let's see if I can pronounce this, Telimogene laherparepvec. Oh my God, it's so bad. TVEC or Imlygic. This was licensed by the FDA in 2015 as Imlygic, and it's injected into melanomas. So what it is, the virus, the herpes virus, remember herpes viruses, you put the gene for GM-CSF in. This stimulates the production of granulocytes and macrophages, which stimulate adaptive immunity against tumor antigens, because remember, adaptive immunity is defective in many tumors. The virus is deleted for a gene called IC34.5 and US11, which causes it to have tumor-specific replication. And then another viral gene, ICP47, is deleted. The product of that gene inhibits antigen presentation. And so this removes that, so there's no inhibition of antigen presentation. And so CTLs can kill the tumor cells. And so this is approved for treatment of melanoma. As I mentioned, myxoma virus is a non-human animal virus that is being used experimentally for treating tumors. It's the same virus introduced into Australia to kill European rabbits, if you remember. It doesn't replicate in any non-rabbit host, but it infects many types of human cancer cells. And we're not sure why. Maybe part of it is that the cancer cells don't have an antiviral response, and the pathways related to transformation in tumors seem to be make them permissive to many viruses. But this is being explored to treat a number of human cancers. Measles virus has also been modified to treat human tumors. They start with the vaccine strain, which preferentially replicates in tumors. This strain cannot antagonize innate immune sensors. And remember, it also binds to CD46, which is overproduced on tumor cells. Also in the vector is the gene for a sodium iodide symporter. What that is, it's a channel that exchanges sodium for iodide. And so during treatment with this virus, of a tumor, gamma-emitting isotopes are given to the patient, which will then get into these tumor cells via this symporter, so you can visualize virus replication in the tumor. And if you give beta-emitting isotopes, they will also get in via this symporter and induce radiation poisoning in the tumor. And this has been used to treat multiple myeloma and is in further trials. And one thing I want you to remember is that therapeutic efficacy of any of these oncolytic viruses is not just lysing cells with the virus, but also activation of anti-tumor responses. So in fact, when a tumor cell is lysed by an oncolytic virus, that activates immune responses. And that seems to be even a bigger part of the efficacy of oncolytic viral therapy than viral lysis of tumor cells itself. And that's illustrated here. Here's your cancer cell. It's infected with your oncolytic virus. And, in many cases, they will lyse tumors. But you can't always infect every cell in a tumor. But what happens is the release of antigens from this tumor will stimulate antigen-presenting cells, which will then educate T cells to come and lyse the tumor cells. And this can kill tumors at distant sites. You can activate these immune responses locally in one tumor, but a patient typically has metastases. There's an effect called the abscopal effect, where you activate local immune responses, and then these activated cells will target tumors at distal sites. And so this is a big research point right now to figure out how we can use this property to do a better job. I think in 10 to 15 years, we're going to be treating many, many cancers with oncolytic viruses such as these and others that remain to be designed. Okay, our last question for today, which is the following statement about oncolytic viruses is incorrect. A, infection by oncolytic viruses leads to destruction of tumor cells only at the site of virus inoculation. B, some viruses of non-human animals can reproduce selectively in human tumor cells. C, viruses with both DNA and RNA genomes can be developed as oncolytic agents. D, various mutations in viral genomes that confer tumor-selective reproduction eliminate or impair viral gene products that counter host interferon defense. Which one is wrong? Your last chance to get 10 correct to 100%, which has never happened before in the history of this course, like 14 years. Okay, we have 5 out of 9 answering. Let's see how we did. So 80% of you got number 1, which is correct. The infection leads to destruction only at the site of inoculation. Remember the abscopal effect. This immune activation can lead to destruction elsewhere. And the only other answer that was picked is number C, viruses with both DNA and RNA genomes can be developed. Absolutely. So measles virus, herpes virus, an RNA and a DNA virus that are being developed. One more application of virus vectors for cancer immunotherapy is CAR T cells. This was FDA approved in 2017 for the treatment of certain blood cancers. And in this, we take advantage of certain antigens that are specific to tumor cells and the fact that T cell responses against cancers tend to be dampened in part because of these checkpoint inhibitors like CTLA4 that I mentioned and PD-L1 that I mentioned before. And what you do is you design a chimeric antigen receptor that will recognize a tumor antigen. And it consists of a part of the T cell receptor, right? The receptor on the T cell that will recognize an antigen on the surface of cells. And then you have made a single chain antibody against that tumor antigen, and you fuse the two into a chimeric antigen receptor. You make these antibodies, and then you clone the genes for them, and you incorporate that into the DNA. You put this in a lentiviral vector. You harvest T cells from the patient. You infect them with the CAR carrying lentivirus, and then you put them back in the patient, and they will enable the T cells to kill tumor cells, and they work very well for certain kinds of blood cancers. So it's another example of how we're using viral vectors to do oncotherapy or cancer immunotherapy. All right. I think this lecture is beautiful in a way that it shows the importance of basic research. Therapeutic viruses that we talked about today have been made possible because of fundamental advances in virology, recombinant DNA, immunology, and clinical science. And serendipity plays a big role in our scientific advances. Some of the things we found today, we discussed today, were discovered as accidents. And so you have to let smart scientists do what they want to do. You can't over-regulate them, because you let them be creative and good things will happen. And so in recent years, there's been a shift towards translational research where you say you cure cancer, you cure diabetes, you cure some other disease, and that's fine. But there has to still be basic research to develop the approaches that can be used. And today's lecture is a perfect example of that. Okay, I want to thank you for taking this course. There aren't many people here, but presumably all of you will listen to it. Here was day one of our virology course way back in January, where everyone was wearing coats. And I would like to thank you and also tell you, don't forget what you have learned here. You know more virology now than 99.9% of the people on Earth. Don't waste it. You don't have to keep it in your head. You just have to remember where to look for it, where to look it up. And I can help you because I'm a public person. I do lots of podcasts, as you know. I'm a big proponent of everyone getting educated. I think education is a key to having a productive life. And I think everybody on the planet should be educated. So I do my part with podcasts on viruses, microbes, parasites, evolution, immunity, neuroscience, infectious diseases, and of course, my virology lectures here. All of this is on YouTube, and you can follow me at any of these places. You have my email for the future. If you ever need to ask a question about some aspect of virology, I'm happy to hear from you. And I always love to hear from you anyway. Tell me what you're doing in your career. And should you ever see a car with viruses in New Jersey, that's me trying to spread the word about viruses.